I've been staring at this stack of research for Chapter 18, and I have to admit, I completely misjudged the characters we are discussing today. Usually when we do a deep dive into bacteria, we're talking about these, rugged, invincible survivors. The spores that can live in the dirt for a century, or the staph that just hangs out on gym equipment waiting to ruin your day.
Right, the tough guys of the microbial world, the ones you can't easily kill with a little bit of heat or drying.
Exactly. But looking at the source material today, we're dealing with a group that seems, frankly, a bit high maintenance.
High maintenance is a very polite way to put it.
Right? These are the gram-negative Coccobacilli, specifically Haemophillis, Bordetella, Brucella, and Francisella. And the text keeps using this one specific word to describe them, fastidious.
Fastidious. It's a great word, isn't it?
It really is.
I mean, in a general sense, it means being attentive to detail or concerned about accuracy. But in clinical microbiology, fastidious is essentially a polite way of saying incredibly picky eater.
Like a toddler who only eats dinosaur-shaped chicken nuggets.
Yes, exactly like that. These are organisms that simply will not grow in the lab unless you cater to their very specific, very complex nutritional demands.
Right. They aren't going to grow on your standard nutrient agar.
Yeah.
They need special vitamins. They need blood cooked in a highly specific way. They need, well, charcoal in some cases. It's a whole production.
It is a massive headache for the lab technicians, yes.
But here's the paradox that I really want us to unravel for you today. How can organisms that are so fragile in the lab, so pathetic, that they literally die if you look at them wrong, be responsible for some of the most terrifying medical emergencies we face?
That is the central question of this entire group. I mean, you have Haemophilus, which used to be the leading cause of meningitis in children.
Right.
You have Bordetella, which causes a cough so violent it can literally break a patient's ribs. You have Brucella, a fever that can last for years. And Francisella, which is actually classified as a potential biological weapon.
So clearly, being fragile in a petri dish does not mean harmless in the human body.
Far from it. Their fastidious nature often means they have evolved to live in very specific niches, usually inside us or inside our animals.
They've traded environmental toughness for the ability to hijack our own biology.
Exactly. They don't need to survive on a park bench because they never intend to be on a park bench.
So our mission today is to decode that hijack. We're going to walk through these four groups, the Coccobacilli squad, if you will, and break down exactly how they get in, why they are so incredibly hard to detect, and how they cause such chaos.
And I think we should start with their shape, since you mentioned the Coccobacilli squad. It's a bit of a morphological middle ground.
Yeah, the text describes them as pleomorphic.
Pleomorphic. So pleo meaning many, and morph meaning shape. When you look at them under a microscope, they can be really confusing.
Because they aren't perfect spheres like cocci.
Right, and they aren't those long, clear rods like bacilli. They are these short, stubby little rods. And depending on how old the culture is or what media they are on, they can stretch out into longer threads or bunch up into tighter spheres.
Which I imagine makes the diagnostic work a total nightmare.
It definitely complicates things. You don't get that clean textbook visual identification immediately. You really have to look at their biochemical behavior to figure out who is who.
Well, let's look at the behavior of the first suspect on our list. Haemophilus.
Haemophilus, the blood lover.
The name itself gives away its obsession, right? Hemo for blood, philus for loving.
It's a very literal description. These bacteria require blood to grow. But it's not just about needing blood in general, it's about why they need it. And this brings us to the most famous member of the family, Haemophilus influenza.
Okay, we have to stop right here and address the elephant in the room, or rather, the virus in the room. Because Haemophilus influenza sounds exactly like it causes the flu.
It does. And for a very long time, the smartest scientists in the world were absolutely convinced that it did.
This goes back to the late 19th century, right? During the great influenza pandemics.
Exactly. Specifically around the 1890 pandemic, there was this very prominent bacteriologist named Richard Pfeiffer. He was isolating this specific bacterium from the lungs of patients who had died of the flu.
And he found it in almost all of them.
He found it everywhere in these autopsies. So logically, he concluded, I have found the cause of influenza, and he proudly named it Bacillus influenza.
But he was entirely wrong.
He was completely wrong. Influenza, as we know now, is a virus. But they wouldn't even discover viruses until decades later. What Pfeiffer was actually seeing was a secondary infection.
The opportunistic strike.
Exactly. The influenza virus ravaged the patient's lungs, destroyed the mucosal defenses, and this opportunistic bacterium Haemophilus just moved into the wreckage to finish the job.
So Haemophilus is basically the guy who walks into the bank after the vault has already been blown open by someone else?
That is a perfect analogy, yes, but the name stuck. The scientific community just kept it. So, for the record, Haemophilus influenza does not cause the flu, but it causes meningitis, epiglottitis, pneumonia, and severe sepsis.
Let's get into those picky eater mechanics I mentioned earlier. The source text spends a lot of time on these two things called X factor and V factor. This feels like the core detective work for the lab. If I'm reading this correctly, Haemophilus literally cannot grow without these two specific ingredients being handed to it.
That is correct. It is an absolute non-negotiable metabolic requirement. And understanding the X and V factors is the only way to understand why microbiologists use the specific agars that they do.
Okay, let's break them down for everyone. Factor X is hemin.
Hemin. It's a heat-stable molecule. Basically, it's an iron-containing porphyrin. Now, most normal bacteria are self-sufficient. They can scavenge iron from the environment and build their own hemin systems for cellular respiration.
But Haemophilus can't.
Haemophilus has lost that genetic ability. It is completely incapable of synthesizing hemin from scratch.
So it has to steal it.
It has to steal it from the host environment, and the richest source of hemin in the human body is hemoglobin, inside our red blood cells.
Okay, so X is hemin, iron. Got it. Now what about factor V?
Factor V is NAD, nicotinamide adenine dinucleotide.
Wow, that brings me right back to college biochemistry. The electron transport chain.
Right. It's a crucial coenzyme. It's essentially the currency of electron transfer in the cell. Again, most organisms can make their own NAD or recycle it efficiently. Haemophilus is terrible at this. It absolutely needs an external supply of NAD to survive.
So here is the puzzle described in the notes, and I found this fascinating. If you take a standard sheep blood agar plate, which is essentially just nutrient jelly with raw sheep blood mixed into it, it is full of red blood cells.
Yeah.
So logically it has hemin, which is X, and it has NAD, which is V, but Haemophilus won't grow on it. Why?
It's a two-part problem. First, the hemin, the factor X, is locked inside the intact red blood cells. These bacteria are small, and they can't necessarily break open those tough sheep blood cells on their own to get the nutrients.
It's like being locked in a grocery store where all the food is in cans, but you don't have a can opener.
Exactly. The food is there, but it's inaccessible.
Okay, so the hemin is locked away. What about the NAD, factor V?
That's the second, more insidious problem. Sheep blood naturally contains enzymes called netadases.
Enzymes that destroy NAD.
Right, they actively break down NAD, so even if a little bit of NAD leaks out of a cell, the plate itself just chews it up and destroys it before the bacteria can absorb it.
So on a standard blood plate, the X is locked up in a vault, and the V is actively being destroyed by enzymes. It's actually a totally hostile environment for this bug.
Precisely, and this is exactly why microbiologists invented chocolate agar.
Which I have to say is incredibly disappointing because it contains absolutely zero chocolate.
No cocoa involved whatsoever, it just gets its name from the color. To make chocolate agar, you take that exact same sheep blood agar and you heat it. You gently cook it to about 80 degrees Celsius.
And the heat solves both of those problems.
Yes, it's a brilliant physical solution. The heat lashes the red blood cells. They pop open, completely spilling their contents, the hemin and the NAD, directly into the agar. The agar turns a brown chocolate color because the hemoglobin oxidizes when heated.
And what about the enzymes destroying the NAD?
Crucially, the heat also denatures those NADase enzymes.
It kills the poison.
It completely destroys the protein structure of the enzyme that would normally eat the NAD. So now you have a plate where all the nutrients are free-floating, pre-digested essentially, and highly stable. It is an absolute buffet for Haemophilus.
That is just such a cool bit of kitchen chemistry applied to microbiology. Now, there is another trick mentioned in the text for when you don't happen to have chocolate agar lying around. These satellite phenomena.
Oh, this is a classic medical board exam question, and it's just a beautiful visual if you ever see it in the lab. Let's say you only have that regular hostile blood agar plate. We just established Haemophilus can't grow on it.
Right.
But if you take a swab of Staphylococcus aureus common staph and you streak a thick line of it right down the middle of that plate.
Just normal skin bacteria.
Right. Staph is a very robust grower. It produces these things called hemolysins, toxins that literally punch holes in red blood cells to get nutrients. So all along that streak of staph, the sheep red blood cells are popping open, and that releases the hemin, factor X.
Okay, so the staph acts as the can opener.
Exactly. And as a bonus, Staph also excretes excess NAD, factor V, as a natural byproduct of its own cellular metabolism. It's essentially sweating out vitamins.
So the staph is providing both the X and the V right into the immediate area.
Exactly. So what you see when you look at the plate the next day is the staph colony growing big and strong right down the middle. And then you see these tiny microscopic dewdrop colonies of Haemophilus growing only right next to the staph line. They are satelliting around the provider.
Like little scavengers just picking up the crumbs falling off the table.
It's commensalism in action right there on a petri dish. If you look even 5 millimeters away from the staph streak, the Haemophilus completely stops growing. It forms this very tight halo.
That is a fantastic image. Okay, so we figured out how to identify it. We know it's incredibly picky. Now let's talk about what actually makes it dangerous to humans. The text focuses heavily on something called the capsule.
The capsule is the single most important virulence factor for Haemophilus influenza, period. If you take a strain and strip away its capsule, the bacteria is relatively manageable. Your immune system can handle it. If it has the capsule, it is a deadly threat.
The text differentiates between encapsulated and unencapsulated strains.
Right. The unencapsulated strains are often called non-typable Haemophilus influenza, or NTI. And these are actually incredibly common. You and I likely have them in our nasopharynx right now. They just live there as part of the normal upper respiratory microbiome.
They're just hanging out.
Mostly, yes. But they can migrate. If they move up the eustachian tube into the ear, they cause otitis media, which are middle ear infections. In fact, they are the second most common cause of bacterial ear infections in young kids, right after pneumococcus. They can also cause sinusitis.
So they cause local annoying infections, mucosal infections.
Exactly. They stay on the surface tissues.
Yeah.
But the encapsulated strains, they are invasive. They don't stay on the surface. And the absolute king of them all is type B.
Haemophilus influenza type B.
Hib. This is the one we fear. Its capsule is made of a very unique biochemical substance. It's called polyribitol ribose phosphate, or PRP.
PRP. Why is this specific sugar structure so incredibly effective at hurting us?
Because it is fiercely antiphagocytic. Think of your white blood cells, your macrophages and neutrophils as the body's internal cleanup crew. Their entire job is to physically grab invading bacteria and eat them.
Right. Phagocytosis.
But the PRP capsule is like a microscopic force field. It's incredibly slick and slippery. The white blood cells physically cannot get a grip on the bacteria to ingest it.
So the bacteria can just float freely in the bloodstream without being eaten.
Correct. And because it isn't being eaten, it multiplies rapidly. It causes severe bacteremia, which is bacteria actively dividing in the blood. And from the blood, it has a highway to go anywhere in the body. The joints, causing septic arthritis, the heart, and most dangerously, it crosses the blood-brain barrier to infect the brain.
Which brings us to meningitis. The text notes that before the vaccine was invented, Hib was the #1 cause of pediatric meningitis.
It's really hard to overstate just how devastating this was. We are talking primarily about very young children, usually between 6 months and 5 years old.
Why that specific age gap?
Because the maternal antibodies they got from their mother during pregnancy wear off around 6 months. But their own infant immune system hasn't fully matured enough to figure out how to attack that slippery PRP capsule yet. It's a window of extreme vulnerability.
And meningitis isn't just a bad headache.
No. It's a rampant infection of the meninges, the delicate lining of the brain and spinal cord. High fever, projectile vomiting, a stiff neck, lethargy progressing to coma. The mortality rate was frighteningly high. And even among the kids who survived the infection, many were left with permanent neurological damage. Severe hearing loss, intellectual disabilities, seizure disorders. It was a true scourge.
There's another clinical condition listed here that I find almost even more terrifying just because of how fast it happens. Epiglottitis.
Oh, epiglottitis is the absolute stuff of nightmares for ER pediatricians.
The text describes the infected epiglottis as cherry red.
Right. So the epiglottis is that little flap of cartilage at the base of your tongue. Its job is to flip down and cover your windpipe every time you swallow, so food goes to your stomach and you don't choke. Haemophilus has a terrifying affinity for infecting this specific anatomical structure.
And when it gets infected, it swells up.
It swells massively and rapidly. It turns into this giant, angry, cherry red ball of inflamed tissue. And the fatal problem is its location. It sits directly on top of the trachea, the airway. If it swells too much, it literally acts like a cork in a bottle. It seals off the airway entirely.
So a child can literally suffocate.
And it happens incredibly fast. A child might go to bed with what just seems like a mild sore throat and a slight fever, and wake up a few hours later gasping for air, sitting up, leaning forward, and drooling because it hurts too much to even swallow their own saliva. The text emphasizes that this is a critical medical emergency. If a doctor suspects epiglottitis, you don't send the kid down the hall to x-ray. You don't lay them flat on their back to examine them because gravity might cause the swelling tissue to fully block the airway. You immediately prep for intubation or an emergency tracheostomy. You have to secure a plastic tube past that swelling before it shuts completely.
It's incredibly intense.
Yeah.
Now, fortunately, we get to talk about the solution to all of this horror, the vaccine. The source material highlights the conjugate vaccine as the sole reason these diseases have basically plummeted in the developed world.
The Hib conjugate vaccine is unarguably one of the greatest public health victories of the 20th century. Invasive Hib cases dropped by over 99% in countries that routinely use it. But the actual immunological science behind how they made the vaccine is what's truly fascinating.
It goes back to that PRP capsule, right? The slippery sugar.
Yes. Here is the massive immunological challenge scientists faced. Sugars, structurally, are terrible antigens.
Meaning they don't trigger a good immune response.
Right. If you take pure PRP sugar and inject it into a human, especially a baby under 2 years old, their immature immune system largely ignores it. It triggers what we call a T-cell independent response. The body might make a few weak antibodies, but it does not form any long-term memory cells.
So the very early attempts to make a vaccine by just injecting the capsule sugar completely failed.
For infants, yes, it worked okay for older kids and adults, but the babies, the ones who were actually dying of meningitis, weren't protected at all. So scientists had to get creative. They developed conjugation.
Which means chemically linking two things together.
Exactly. They took the weak PRP sugar and they chemically covalently bonded it to a very strong protein carrier. The text mentions they often use diphtheria toxoid, or a specific meningococcal outer membrane protein.
Why add the bacterial protein to the sugar?
Because the infant immune system absolutely loves to attack foreign proteins. It's evolutionarily very good at spotting them and mounting a massive response. So the baby's immune cell sees this diphtheria protein, grabs it, pulls it inside to process it, and in the process it accidentally pulls the attached PRP sugar in along with it.
It's basically a Trojan horse, but in reverse. You use the dangerous-looking protein to sneak the invisible sugar into the immune system's education center.
That is exactly what it is. By processing the protein, the infant's immune system is forced to present the attached sugar to the T cells. The body essentially says, oh wait, this slippery sugar is physically attached to this dangerous diphtheria protein. I better memorize the sugar structure too, just to be safe.
And boom, you suddenly get long-term immunity and memory B cells against a sugar.
A robust T-cell dependent memory response. And that's why we vaccinate infants at 2, 4, and 6 months of age. We can actively protect them during that vulnerable window now, whereas before, parents and doctors just had to hold their breath and hope the child didn't catch it.
That is absolutely brilliant engineering. Now, before we leave the Haemophilus group entirely, I want to quickly touch on the specific cousins mentioned in the chapter. H. aegyptius.
Ah, yes. This is a really interesting one. Historically, it's just associated with pink eye, a very contagious but generally harmless purulent conjunctivitis. But specifically in Brazil, back in the 1980s, a new genetic clone of this bacteria emerged that caused something called Brazilian purpuric fever.
That sounds incredibly ominous.
It was devastating. The infection starts exactly like normal pink eye, but instead of staying in the eye, it suddenly invades the bloodstream. It rapidly progresses to high fever, severe purpura, which is widespread purple bleeding underneath the skin, and ultimately fatal septic shock. It killed a lot of children very quickly.
Why did it change?
It acquired a new virulence plasmid. It just perfectly illustrates how dynamic these fastidious bacteria can be. They can evolve. A relatively annoying, common eye infection mutated into a systemic killer almost overnight.
Wow. And the other one mentioned is H. ducreyi. This one is an STD.
Yes, chancroid. It's much more common in tropical and subtropical regions. The key diagnostic takeaway here for clinicians is distinguishing it from the ulcer caused by syphilis.
The text calls it the soft chancre.
Right. Syphilis causes a hard, indurated, completely painless ulcer on the genitals. H. ducreyi, on the other hand, causes a soft, ragged, and extremely painful ulcer. And the local lymph nodes in the groin swell up massively and can actually spontaneously rupture and drain pus.
Painful versus painless. That's the rapid diagnostic key.
Correct. Painful means think ducreyi. Painless means think syphilis.
Okay, that comprehensively wraps up Haemophilus. Let's move down the respiratory tract to the second major group on our deep dive.
Bordetella.
Bordetella pertussis, the notorious agent of whooping cough.
This one is also a extremely small gram-negative coccobacillus. It also has a capsule to protect it. But growing it in the lab seems even more bizarrely specific than Haemophilus. The text mentions the classic Bordet Gengu medium. Yes, this is real old-school microbiology from the early 1900s. The Bordet Gengu medium is made of potato infusion, blood, and glycerol.
Potato. Why on earth is there potato in a medical lab test?
It does sound like a strange soup recipe. But the potato infusion is chemically crucial. It provides a massive amount of starch. You see, Bordetella is incredibly sensitive to environmental toxins, specifically fatty acids, heavy metals, and sulfides. If you try to put it on regular lab agar, the microscopic trace toxins naturally present in the agar itself will instantly kill the bacteria.
So it's poisoned by its own petri dish.
Exactly. The complex starch from the potato acts like a chemical sponge. It absorbs and neutralizes all those trace toxic substances in the agar.
It acts like a protective filter.
A detoxifier. It creates a chemically safe space for the bacteria to slowly multiply. And when it finally does grow on this medium, the colonies look very distinct. The text describes them as looking like tiny drops of mercury or bisected pearls resting on the agar.
There's also a newer growth medium mentioned, Regan-Low.
Yes, Regan-Low is the modern standard. It uses charcoal instead of potato, but it's the exact same chemical principle. The activated charcoal strongly absorbs the toxic fatty acids and peroxides so the delicate bacteria can survive. Again, it highlights the fastidious nature. It absolutely cannot handle the real world without intense protection.
Let's get to the disease itself, whooping cough. I think many people today view this as a historical nuisance, something from Victorian novels. But when you read the pathogenesis section of this text, it is a highly coordinated, violent biological assault on the body.
It is essentially a microscopic toxin factory. The bacteria doesn't just quietly invade. It fundamentally disrupts the entire physiology of the human lung. And this entire assault is coordinated by a master genetic switch called the BVG gene.
The BVG locus.
It stands for Bordetella virulence gene. It's a complex environmental sensor. When the bacteria is floating in the air or on a surface, the temperature is low, the nutrients are low, and the BVG genes are firmly switched off. It's in survival mode. But when it gets inhaled and senses, it's inside a warm human airway at exactly 37 degrees Celsius.
It flips the switch.
It flips the switch, and it instantly starts pumping out a highly orchestrated sequence of binding proteins and destructive toxins.
Let's walk through that attack sequence. Step one has to be attachment, right?
Right. Bordetella is fundamentally a non-invasive surface pathogen. Unlike Haemophilus, it doesn't usually burrow into the tissue or go into the blood. It strictly sits on the very surface of the airway lining. To stay there, it uses a massive surface protein called filamentous hemagglutinin.
Which essentially acts like a grappling hook.
Exactly, a highly specific grappling hook. It binds exclusively to the cilia of the respiratory epithelial cells.
Okay, we really need to pause on the cilia, because these tiny structures are the unsung heroes of the lung and the main victims here.
They absolutely are. Your entire lower respiratory tract is lined with millions of these microscopic hair-like cellular structures called cilia. And they beat constantly in a perfectly coordinated upward rhythm. They're constantly sweeping a layer of mucus, trapped dust, dead cells, and inhaled bacteria up and out of your lungs toward your throat to be swallowed.
It's the self-cleaning oven function of the lungs. The text calls it the mucociliary escalator.
Precisely. It is a vital defense mechanism, and Bordetella targets them with pinpoint accuracy. Once it latches onto the cilia with its grappling hook, it begins releasing its first major weapon, tracheal cytotoxin.
And what exactly does this cytotoxin do?
It is a piece of bacterial cell wall that specifically kills the ciliated cells. It enters the cell, completely inhibits their DNA synthesis, and paralyzes the physical beating mechanism of the cilia. Eventually, the ciliated cells just extrude from the airway lining and die.
So the escalator just stops working.
The escalator violently breaks down. But your lungs are still naturally producing mucus. In fact, they are producing way more mucus now because of the severe inflammation of the infection. So you have this thick, sticky, infected mucus rapidly accumulating deep in the airways and absolutely no mechanical way to sweep it out.
And that buildup is the physical setup for the infamous cough.
The cough is the body's desperate last resort. Since the gentle, constant sweeping mechanism is totally dead, the body has to use the nuclear option to clear the airway. It uses violent, explosive, mechanically damaging bursts of air to try and dislodge that concrete-like sludge.
This leads perfectly into the clinical progression. The text breaks the disease into 3 stages, and the second one, the paroxysmal stage, is where that cough happens. I was reading the clinical description of these specific coughing fits, and it just sounds physically exhausting.
Exhausting is a massive understatement. A paroxysm is a sudden, violent attack. A patient, usually a young child, might cough 15 to 20 times rapidly in a single, continuous exhalation. They are literally forcing every single milliliter of reserve air out of their crushed lungs.
And that extreme exhalation is what creates the whoop.
The whoop comes right after the coughing fit ends. The patient has emptied their lungs completely. They're severely oxygen-starved. They need to inhale a massive volume of air immediately just to survive. But the vocal cords and the airway are incredibly swollen and inflamed from the trauma of the cough, so they inhale desperately against a tightly narrowed glottis.
It's the physical sound of extreme air turbulence.
Yeah, exactly. It's the terrifying sound of someone gasping for their life through a tiny, swollen straw, and that high-pitched, desperate intake of air is the characteristic whoop.
And the text makes a critical note that very young infants often don't actually whoop.
They don't. They physically don't have the muscular chest strength or the developed airway dynamics to generate that massive negative pressure. Instead of whooping, they have profound apnea. They have a coughing fit, and then they just stop breathing entirely. They turn blue, which is cyanosis. They can suffer seizures from the lack of oxygen to the brain. That is exactly why pertussis is so incredibly deadly in babies under 6 months old.
Now, there are other toxins involved in this process too, right? Pertussis toxin itself?
Yes, pertussis toxin, or PT, that's the major systemic one. While the bacteria stays in the airway, PT gets absorbed into the patient's bloodstream. And it does a lot of complex cellular disruption, but one of its weirdest, most textbook clinical effects is profound lymphocytosis.
Meaning an abnormally high white blood cell count.
Extremely high, and a very specific type. Usually in a classic bacterial infection, you see a spike in neutrophils. But in pertussis, you see a massive spike in lymphocytes, which are the immune cells that usually fight viruses. The toxin actively blocks the lymphocytes from leaving the bloodstream to enter the lymph nodes, so they just pool in the blood. It completely confuses and derails the normal cellular immune response.
So you have a paralyzed lung cleaning system, a chemically confused immune system, and a patient who is coughing so hard they vomit, burst blood vessels in their eyes, or literally fracture their own ribs.
Exactly. It is a brutal disease, and the recovery is agonizingly slow. The third stage is called the convalescent stage. It can take months for those specialized ciliated cells to fully grow back and restore the escalator.
That's why the traditional Chinese medical name for it translates to the 100-day course.
Right, because even long after your immune system or antibiotics have killed every single Bordetella bacteria, the physical architectural damage to the lung remains. You keep coughing for weeks just trying to clear normal daily mucus.
Let's shift to prevention, because we do have a vaccine for this.
Right.
But the historical story of the pertussis vaccine is surprisingly complicated. We actually completely switched vaccine types a few decades ago, didn't we?
We did. We started back in the 1940s with the whole cell vaccine. It was exactly what it sounds like. They literally grew vats of Bordetella pertussis, killed the bacteria with heat or chemicals, ground them up, and injected the whole mess.
And it worked.
It worked incredibly well. It drove pertussis numbers down to almost zero. The immune system saw every single part of the bacteria and mounted a fierce, long-lasting defense. But it was highly reactogenic.
Meaning it had a lot of noticeable side effects.
Yes. Because it contained all the endotoxins and cellular debris of the bacteria, babies often got very high fevers, extremely swollen, painful arms at the injection site, and prolonged, inconsolable crying fits. Understandably, parents were terrified. There were massive public campaigns and lawsuits claiming the vaccine caused permanent neurological damage, even though massive subsequent studies showed no actual causal link to encephalopathy.
But the public pressure and fear were intense enough that science had to pivot.
Exactly. The fear of the vaccine became worse than the memory of the disease. So scientists developed the acellular vaccine, denoted by a little a in DTaP.
Which contains just specific purified parts of the bacteria.
Right. They just purified the key antigens, the pertussis toxoid, the filamentous hemagglutinin, and a few others. It is chemically much cleaner. And it worked. It causes drastically fewer fevers and local reactions.
But there's a massive catch, isn't there?
There is always a trade-off. Because the immune system is only seeing a few purified proteins instead of the whole complex organism, the resulting immunity isn't nearly as robust, and crucially, it is not as long-lasting. With the old whole-cell vaccine, protection lasted a very long time. With the newer cellular vaccine, clinical protection seems to actively wane after about 5 to 10 years.
Which perfectly explains why we constantly see these outbreaks in middle schools and high schools now.
Exactly. These teenagers were fully vaccinated as kids, but their cellular protection has slowly faded. So they catch the bacteria. Now, they don't usually get the classic rib-breaking whooping cough. They usually just get a persistent, annoying cough for a few weeks. But while they are coughing,
They are shedding the bacteria everywhere, and they can spread it to a newborn sibling who hasn't been vaccinated yet.
precisely. And that dangerous transmission gap led public health officials to develop the concept of cocooning.
Cocooning, I like the imagery of that. What does it actually mean in practice?
Since we physically cannot give the pertussis vaccine to a newborn immediately, their immune system just won't respond to it, yet we have to build a protective barrier around them. So we aggressively vaccinate everyone who will come into contact with the baby. The pregnant mother gets a booster in her 3rd trimester to pass antibodies through the placenta. The father gets a booster. The grandparents, the babysitters. You actively build a cocoon of immunity around the vulnerable infant to block the transmission chain.
It's a very clever strategy born entirely out of the biological limitations of our current acellular vaccine.
Exactly. It's a stopgap measure until we can design a better vaccine.
Okay, we've thoroughly covered the throat and the lungs. Now let's entirely leave the human respiratory tract for a moment and take a trip to the farm. Let's talk about the third group, Brucella.
The Brucella group. These are the classic examples of zoonoses, which are infectious diseases that naturally jump from vertebrate animals to humans.
The text lists 4 main species of Brucella, and they seem to have very specific, rigid preferences for their animal hosts.
It's very much like a pathogen menu. Brucella abortus strongly prefers cattle. It causes infectious abortion in cows. Brucella sus prefers domestic and wild pigs. Brucella canis naturally prefers dogs. And Brucella militensis strongly prefers goats and sheep.
And for humans catching the disease, melitensis is the one we really have to worry about.
They can all cross over and infect humans if given the chance. But B. melitensis, the goat and sheep strain, is by far the most virulent and causes the most severe prolonged systemic disease in human patients.
How exactly do we usually catch this? I assume the average person isn't wrestling infected pigs or assisting in sheep births on a daily basis.
Historically, and honestly still in many parts of the world today, the primary transmission route is foodborne. Specifically, the ingestion of unpasteurized dairy products, milk and cheese.
Ah, the raw milk trend.
It is an incredibly dangerous culinary trend. You see, Brucella loves to actively concentrate and multiply in the mammary glands of infected female animals. So the raw milk coming from that animal is absolutely teeming with live, virulent bacteria. If you eat soft, fresh goat cheese or drink milk, particularly from regions where the disease is endemic, like the Mediterranean basin, parts of the Middle East, or Mexico, and it hasn't been pasteurized by heat, you are taking a massive microbiological gamble.
But there's also significant occupational risk mentioned for certain professions.
Yes, direct contact transmission. Farmers, large animal veterinarians, abattoir workers. If a vet is manually helping a cow deliver a calf that is infected with Brucella, or a worker is handling raw infected meat, the bacteria can easily enter the human body through microscopic cuts or abrasions in the skin. Or, very commonly, infectious fluids can be splashed directly into the mucous membranes of the eyes.
Once it actually gets inside the human body, Brucella pulls a really sophisticated, almost sci-fi trick. The text calls it a facultative intracellular parasite.
This is its absolute evolutionary superpower. Normally, when a foreign bacteria enters human tissue, it is immediately hunted down by patrolling macrophages. The macrophage engulfs the bacteria, swallows it into a little cellular bubble called a phagosome, and then aggressively dumps digestive acid and toxic enzymes into that bubble to dissolve the invader.
Standard immune defense. But Brucella gets eaten, and it intentionally doesn't die.
It completely hacks the macrophage's internal machinery. It secretes specific proteins that actively prevent the macrophage from fusing the acid-filled lysosomes with the bubble it's trapped in. So instead of being a prisoner locked in an acid bath, Brucella essentially turns the macrophage into a luxury hotel room. It happily lives and rapidly multiplies right inside the macrophage.
And because macrophages are immune cells that naturally travel around the body.
The Brucella just takes a free ride. It uses our own immune cells as a taxi service. It travels from the initial site of infection straight to the regional lymph nodes. And from there, it dumps into the bloodstream and seeds all the major filtering organs. The liver, the spleen, the bone marrow. It spreads systemically while literally hiding inside the very police cars that were dispatched to destroy it.
That is terrifyingly smart. And how does the human body eventually react to this internal invasion? The text mentions the formation of granulomas.
Right. The human immune system isn't totally helpless. Eventually, the T cells realize that these macrophages are infected and compromised. But since they can't easily kill the tough bacteria hiding inside the cells, the immune system decides to just quarantine the entire affected area. It actively builds a physical wall. It tightly packs thousands of immune cells around the infected tissue, creating a hard nodule called a granuloma.
To physically wall off the infection.
Exactly. And because the body is building thousands of these microscopic granulomas inside the organs, it causes those organs to physically swell. You get profound hepatosplenomegaly, which is a massively enlarged liver and spleen.
And what does this actually feel like for the patient? The classic clinical symptom listed is undulant fever.
Undulant essentially means waving or rising and falling like a wave. The fever in brucellosis isn't a constant, steady high temperature like you see in other infections. It follows a brutal daily cycle. The patient's temperature slowly rises during the afternoon, peaks very high in the late evening, and then dramatically breaks in the middle of the night with absolutely profuse, drenching sweating. Then they wake up feeling exhausted but relatively normal in the morning, only for the entire cycle to start over again that afternoon. And this can go on for weeks or even months.
The text mentions a very specific, bizarre detail about the smell associated with that night sweat.
Yes. The sweat of a brucellosis patient is said to smell distinctly like wet hay.
Wet hay.
A very strong, moldy, slightly sweet barnyard smell. It's highly distinctive for clinicians who know what to look for, combined with the undulating fever, profound chronic fatigue, and often very deep, severe bone and joint pain because the bacteria loves to infect the spine and cause osteomyelitis.
Diagnosis of this disease seems incredibly tricky. The text explicitly warns about massive lab safety issues when trying to culture it.
Brucella is notoriously one of the most common causes of laboratory-acquired infections in the world. It is highly, highly infectious via the aerosol route.
Meaning just breathing it in.
Right. In the old days, a common way a lab tech would do a quick check to see if a culture was growing was to pop the lid off the Petri dish and physically sniff it to smell the bacterial byproducts. If a tech opens a plate of Brucella on an open benchtop and sniffs it, they will almost certainly inhale enough microscopic aerosolized bacteria to contract severe brucellosis. It must be handled in a strict biosafety cabinet.
So absolutely do not sniff the Petri plates.
Never, ever sniff the plates. But because blood cultures are so slow and frankly dangerous to grow, we primarily rely on serology for diagnosis. We draw the patient's blood and look for anti-Brucella antibodies. And here we hit a massive mathematical weirdness in the lab called the prozone phenomenon.
Okay, I read this section 3 times and I'm still trying to wrap my head around it. It essentially says that if a patient is super infected and has too much antibody in their blood, the diagnostic test will actually fail and say they're negative.
It sounds completely contradictory, but yes, it does. You have to think about the physical mechanics of how an agglutination test actually works on the slide.
Okay, break it down for me.
You have a reagent, which is dead Brucella bacteria, the antigen, and you add a drop of the patient's blood serum, which hopefully contains antibodies. The structure of an antibody looks like the letter Y. It has 2 sticky arms. The goal of the test is for one arm of an antibody to grab onto bacteria A and the other arm to reach out and grab onto bacteria B. This physically links the 2 bacteria together.
Right, and if millions of them link together, they form a massive clump that you can see with your naked eye. Clumping equals positive test.
Exactly, cross-linking. But if the patient has a chronic, severe infection, they might have a massive, overwhelming concentration of antibodies in their serum. When you drop that super concentrated serum onto the bacteria, every single available binding site on every single bacteria gets instantly coated by its own individual antibody.
It gets swarmed.
Swarmed. There's physically no open space left on any bacteria for an antibody to reach across and grab a second one. Every Y-shaped antibody is just holding on to one bacteria, waving its other arm in the empty space.
They were too crowded to hold hands, essentially.
That is a perfect way to visualize it. They are completely coated, but they are not connected to each other, so the bacteria do not clump together. The solution remains smooth, and the lab tech looks at it and writes down, negative for Brucella.
So you have a patient who is clearly severely sick, loaded with antibodies, but the test falsely says no. How do you fix that?
To fix the prozone effect, you literally have to dilute the patient's serum. You add sterile saline water to it. You systematically reduce the sheer number of antibodies in the tube until they are sparse enough that they are forced to start reaching out and cross-linking the bacteria again.
You have to mathematically dilute the sample just to be able to see the mass of infection. That is incredibly counterintuitive.
It is. It's all about finding the zone of equivalence. You need the precise thermodynamic ratio of antigen to antibody for clumping to visually occur. If a doctor strongly suspects brucellosis and the initial test is negative, they have to specifically instruct the lab to run serial dilutions to check for that prozone phenomenon.
Treatment for this sounds incredibly difficult, too, given the hiding spots we talked about.
It's a therapeutic nightmare because the bacteria hide deep inside the macrophages and inside those hard granulomas. Standard antibiotics floating in the bloodstream often can't reach them. You have to use specialized antibiotics that can actively penetrate the human cell membrane and work in that acidic intracellular environment. The standard gold treatment is usually a combination therapy of doxycycline plus rifampin. And you have to treat the patient for a very long time, a minimum of 6 full weeks, sometimes much longer, just to slowly dig out and kill every last hidden bacteria to prevent a relapse.
And prevention, since this is zoonosis.
The single most effective public health intervention is the mandatory pasteurization of all commercial milk and dairy products. It's really that simple. Heat kills Brucella instantly. The second pillar is aggressively vaccinating the cattle herds. We have a very effective live attenuated vaccine for cows called Strain 19, but we do not have a safe vaccine for humans, so we protect the humans by managing the animal reservoir.
Okay, we have one final, incredibly intense member of the Coccobacilli squad left. And this one feels by far the most dangerous in terms of raw power and intentional human threat. Francisella tularensis.
The dreaded agent of tularemia, which is historically also known as rabbit fever or deer fly fever.
And currently classified by the government as a Tier 1 select agent. That legally puts it on the bioterrorism watch list alongside things like anthrax and Ebola. Why is this tiny bacteria so heavily guarded?
It comes entirely down to its terrifying biological efficiency. In infectious disease, we measure the relative danger of a pathogen by its infectious dose. That is the minimum number of individual bacterial cells you need to accidentally inhale or ingest to establish a fatal infection.
For something like salmonella food poisoning, it's usually in the hundreds of thousands or even millions of cells, right?
Right. You have to eat a heavily contaminated piece of chicken to get sick. For Francisella tularensis, the infectious dose is incredibly, shockingly low. 10 to 50 individual organisms.
10 in individual cells. That is essentially invisible.
It is microscopically small. It's nothing. You could easily fit enough Francisella bacteria on the head of a single pin to lethally infect an entire army battalion. That extreme infectivity is exactly why both the United States and the Soviet Union aggressively weaponized and stockpiled it during the Cold War. It can be easily aerosolized into a massive invisible cloud. It's highly environmentally hardy for a fastidious organism, and it causes rapid, incapacitating, and often fatal illness in almost everyone exposed.
But in the natural everyday world, we aren't usually dodging Cold War bioweapons. We're dodging wild rabbits and infected ticks in the woods.
Exactly. Outside of a lab, it is strictly a zoonotic disease of wild animals. It naturally circulates in huge numbers among wild rabbits, hares, muskrats, and various rodents. And it is primarily transmitted between these animals and accidentally to humans by the bites of infected hard ticks, primarily the dermacenter species, or biting deer flies.
The source text describes several totally different clinical forms of tularemia, and it seemed to depend entirely on how the bacteria physically entered your body. The most common natural form listed is ulceroglandular.
This is often classically referred to as the hunter's disease. Imagine you are out in the woods, you just shot a wild rabbit, and you are skinning it with a hunting knife. You happen to have a tiny microscopic paper cut or nick on your index finger.
And the infected blood gets on your hand.
The heavily infected rabbit blood gets smeared over that tiny nick. Those 10 to 50 bacteria easily slip right into your dermis.
What happens next?
Within a few days, you develop a very nasty black necrotic ulcer directly at the exact site of entry on your finger. It literally punches a painful hole in your skin. And then the bacteria rapidly travel up the local lymphatic system to the regional draining lymph nodes. Usually for a hand injury, that means the lymph nodes in your armpit. Those nodes swell up massively, becoming incredibly painful and fluctuant.
It sounds horrifyingly like the bubonic plague.
The clinical presentation is actually strikingly similar to plague buboes. Huge, tender, swollen lymph nodes that can eventually necrose and drain pus through the skin. It's a severe, high-fever systemic illness.
There's also another entry route called oculoglandular.
That happens if you are handling that same infected rabbit. You get the blood on your fingers, and before you wash your hands, you absentmindedly rub your eye. The bacteria instantly penetrates the conjunctiva. You get severe, painful, purulent conjunctivitis, and the lymph nodes right in front of your ear and down your neck swell up massively.
But the specific form that directly relates back to that bioweapon potential we mentioned and also presents a truly bizarre, very specific suburban hazard is pneumonic tularemia.
Yes, this is what happens when you physically breathe the bacteria straight deep into your lungs. And the textbook gives a very specific and honestly somewhat gruesome epidemiological example of how this happens naturally to regular people. Lawn mowing.
Lawn mowing, seriously.
Seriously, imagine a wild rabbit that has died of tularemia, or even a nest of infected baby rabbits hidden deep in tall grass. A homeowner is out doing yard work and pushes a high-powered rotary lawnmower right over that nest.
Oh no, oh that's awful.
It is grim. But from a physics perspective, those spinning mower blades essentially act as a massive mechanical aerosolizer. The machine instantly pulverizes the highly infected animal tissue and sprays a massive invisible cloud of biological aerosol straight up into the air, right into the breathing zone of the person pushing the mower.
And because we established the infectious dose is only 10 microscopic organisms.
They breathe that invisible cloud in, and 3 to 5 days later, they develop sudden onset, explosive, highly atypical pneumonia, extreme fever, chest pain, coughing, and rapid respiratory decline. It has a very high mortality rate if it is not immediately treated, and it is an incredibly difficult diagnostic challenge for the ER doctor because no one is naturally looking for exotic rabbit fever in a suburban dad who just came in with a cough after doing his Saturday yard work.
Diagnosis of this, again, relies almost entirely on serology because trying to culture in the lab is a death wish.
Yes, it actually requires a very specific, highly enriched media containing the amino acid cysteine to grow at all, but most standard hospital microbiology labs will absolutely refuse to even attempt to culture it if they suspect tularemia. The aerosol risk to the laboratory staff is just far too high. The moment the lab suspects Francisella, they seal the plates, lock them down, and immediately ship the samples to a high-containment state health department lab or the CDC. So for rapid local diagnosis, doctors rely on blood agglutination tests to look for the specific antibodies. But there is a massive diagnostic catch here, too.
Cross-reactivity, the ghost of our previous pathogen.
Exactly. The surface antigens of Francisella tularensis and Brucella abortus are biochemically very similar. They share structural epitopes, so the antibodies your body makes against one can physically bind to the other in the lab test. A positive antibody test for Francisella might actually be a false positive, and the patient really has a Brucella infection, or vice versa.
So the doctor literally has to play detective and ask the patient about their weekend. Did you recently eat any unpasteurized goat cheese imported from Mexico? Or did you accidentally run over a rabbit with your lawnmower?
Context and a thorough patient history are absolutely everything in diagnosing these fastidious zoonoses. The lab test alone is not enough. You have to know the epidemiological exposure.
And how do we actually treat Francisella if we catch it in time?
The classic gold standard drug of choice is streptomycin. It's an older, very powerful aminoglycoside antibiotic that rapidly kills the bacteria. Gentamicin works very well too. Doctors sometimes try to use oral tetracyclines, like doxycycline, because they are easier to take.
Yeah.
But tetracyclines are only bacteriostatic. They just pause the bacteria from growing. They don't actively murder them. Because Francisella is another sneaky intracellular pathogen that hides inside macrophages, if you just pause it with doxycycline, the moment you stop the pills, the bacteria wakes back up and the patient suffers a severe clinical relapse. You really need the heavy-hitting bactericidal drugs like streptomycin to clear it completely.
Wow. We have really covered a massive amount of microbiological ground here today. We've gone from the devastating throat infections of Haemophillis to the paralyzed lungs of Bordetella to the agricultural hazards of Brucella and finally to the weaponized rabbit nests of Francisella.
It is an incredibly diverse and clinically challenging group of organisms.
Yeah.
Despite sharing that simple Coccobacilli shape, their disease profiles couldn't be more different.
If you had to conceptually synthesize all of this, if there is a single defining thread tightly connecting these four pathogens, what is the main takeaway for you?
For me, looking at all of them, the central unifying theme is extreme evolutionary specialization. These organisms are not rugged generalists. They don't live everywhere. They have evolved over millions of years to perfectly exploit very specific, very tiny weaknesses in the human host's armor.
They are biochemical lockpicks.
Exactly. Haemophilus specifically exploits our constant biological need for iron and our infants' inability to defend against slippery sugar capsules. Bordetella flawlessly exploits and dismantles the very physical escalator mechanism designed to keep our lungs clean. Brucella subverts the very macrophage cells dispatched to kill it, turning them into protective transport vehicles. And Francisella exploits our extreme vulnerability to incredibly low-dose aerosol exposures.
And because they are so highly rigidly specialized to exploit us, they are fastidious. They physically cannot survive without us.
They absolutely need the host. And that extreme intimacy is exactly what makes them so dangerous. But, crucially, it also makes them targetable by modern medicine. Because they are so rigid, we can learn their specific weaknesses. We finally figured out that Haemophilus relies entirely on that PRP sugar capsule for survival, so we biochemically engineered a conjugate vaccine to attack that exact sugar. We discovered that Bordetella uses specific binding proteins and toxins, so we purified those exact pieces to create the acellular pertussis vaccine.
So detailed scientific knowledge is literally the weapon.
Absolutely. The deeper we dive into their fastidious, specialized biology, the better we can dismantle them.
I really want to leave you all with one final, slightly provocative thought to mull over. We talked at length about Bordetella pertussis, the whooping cough, and the current massive problem of waning immunity with our modern cellular vaccine. It really feels like we are locked in a continuous, high-stakes arms race with these organisms. We developed a much safer vaccine, which was a great triumph, but the bacteria essentially seems to be finding a way to exploit that safety, or at least just quietly waiting us out until our temporary immunity fades in high school. Are we potentially going to see a massive, widespread resurgence of these supposedly conquered Victorian diseases simply because our modern medical tools, while safer, are actually less durable?
It is a very real, very pressing concern in public health right now. Microbial evolution absolutely never stops. As we constantly change our vaccine strategies, the natural selection pressure we put on the bacteria dramatically changes. We are forcing them to adapt. We very likely might need entirely new biochemical strategies, or at the very least, entirely new schedule regimens for boosters to ensure we keep the 100-day cough permanently in the history books where it belongs.
Well, on that slightly ominous but highly scientific note, wash your hands thoroughly, please cook your meat to temperature, strictly pasteurize your fancy cheeses, and for everyone's sake, please carefully check the tall grass for rabbit nests before you start the lawnmower this weekend.
Very sound, highly practical advice.
Thanks for diving deep into the microscopic world with us today. We'll see you next time.